الله مساءكم. أسعد الله مساءكم بكل خير، وسعيدة بهاللقاء. إن شاء الله يا رب اليوم تتحقق الفائدة المرجوة والجميع يستفيد بأكبر قدر ممكن. دائما نفضل أنه إحنا يعني اليوم ودي يعني أتعرف على الباك جراوند الحضور من فضلكم إذا سمحتوا لي، بحيث أننا نحاول نتبادل اكسبيرينس والفائدة بقدر المستطاع. أنا أعرف وأتوقع أنه أغلبكم باحثين دكتوراه، فاحتياجكم أكثر للفهم والمعلومة بشكل دقيق أكبر من المعلومة بشكل عام. فيهمني جدا أتأكد من نسبة طلبة الدكتوراه اليوم. ودي أسمع صوتكم، ودي يعني يصير في نقاش، بس أفضل بعد كل سيشن راح نتناقش فيه. اليوم إن شاء الله راح يكون موضوع سموث، راح يكون مبسط جدا، ما راح ندخل في ديتيلز يعني أو ما راح نتعمق في كل الموليكولر جينيتكس. راح يكون عندنا 2 parts إن شاء الله، وأتمنى أنه يكون إن شاء الله مفيد. يا ليت أسمع منكم وأعرف يعني الحضور باك جراوند قبل ما نبدأ. ممكن أستاذ ريم يعني تسمحي لهم نسمع أصواتهم؟
تمام. حابة صوت مو
أي صوت أفضل عشان
تمام. إذا في أحد حاب يشارك بس يرفع يده. أصدقاء لندن، سمعونا أصواتكم.
أنا أتمنى أن صوتي مسموع.
لا، واضح صوتك تمام.
ممتاز ممتاز.
طب نسأل، معانا طلاب دكتوراه اليوم؟ ياك مروة، هلا والله.
طيب، أهلا وسهلا بالجميع عموما. طيب، راح نخش في الموضوع على طول. اليوم I would like to start to present a case to investigate. Then we will go through how can we diagnose this case. Nora, she's a lady, 55 years old, present to the emergency department in our hospital following 2 weeks of progressive dyspnea and fatigue and a new rash. During the physical exam, the doctor will likely pay close attention to her eyes, mouth, skin, lymph node, liver, spleen, and nervous system, and he will look for the areas of bleeding or bruising or possible signs of infection. The physical examination results are within the normal limits, except for skin pale and partial rash on the lower extremities bilaterally. The doctor immediately requests a blood test as usual. If there is reason to think there might be problem caused by the low level of blood cells, either due to anemia, friction, bleeding, bruising, etc., or the doctor you most likely will order a blood test to check her blood cell count and the biochemistry. Here, CBC result showed there is a significant increase in the WBC count. There is low count of platelets, significant increase or critical increase of ESR associated with low hemoglobin and RBC count. So the patient had no previous hematology or oncology history, no previous CBC received at our lab. The critical WBC was called to the physician in the emergency, and based on WBC flagged auto differential, make a slide to immediately to send it for television or microscope bench. On a television or microscopic bench, that showed there is a rare neutrophil was seen in the peripheral smear with immature appearing monocyte, few lymphocytes, and also many blasts can be seen. It's about 88% of blast can be seen under the microscope. The pathologist reviewed the slide and the sample was sent for the flow cytometry studies. The pathologist also commented that there is a numerous blast consist with acute myeloid leukemia feature. Specimen to be submitted for flow cytometry, and hematopathologist consultant recommended and was added to the report. The myeloid lymphoid disorder and acute leukemia analysis by the flow cytometry showed there is a myeloid positive markers for CD117, CD33, and CD13, as you can see in the bottom histograms. So the pathologist requests a bone marrow biopsy for this patient for morphology and for cytogenetic studies and FISH. Bone marrow biopsy reveals there is hypercellular marrow with 60% blast consist with a diagnosis of acute myeloid leukemia. However, the cytogenetic report showed there is no demonstration of 1517 translocation, and there is no evidence for PMRR alpha gene rearrangement, translocation 1821, no evidence for MLL gene locus, no evidence for inversion 16 or Pcr ABL rearrangement, as can we see in FISH result in your right-hand side. So the molecular diagnosis or molecular test has been required now to investigate markers for AML. The doctor or the hematopathologist, he requests FLT3, IDH, IDH2, and PM1 genes to be investigated as the main markers or the most markers for a diagnosis of acute myeloid leukemia. So the aim of our lecture today is to identify the applications of molecular genetics technique and different type of mutations are for genetic diagnosis of the genetic disease. Before we go through this, we need to understand or we need to know the sources of these mutations or that cause DNA damage and how the cell responds for this kind of damage. The damage in the cells, it's either, if it happens in the cells, it's either intrinsic due to the spontaneous base modification or during the replication process that really lead to replication error, or maybe the source of damage due to exposure to the chemical agents, either exogenous, that for example, any chemicals mutagens, or endogenous during the metabolic process inside the cells, which produce oxygen radicals. Or it might be radiation, is exposure to the ultraviolet radiation or ionizing radiation. If the DNA or your cells expose any kind of these sources of DNA damage, that will lead to produce DNA lesion. The lesions, there is a variety of lesions can be produced, either base pair mismatch, it could be lead to produce insertion in the DNA, deletions of DNA or double-stranded breaks. There is cross-link strands of the DNA. Sorry. However, based on this, the damage or severity of the damage, the cell will respond to the damage. There is two kinds of damage. If there is the damage, the amount of the damage can be handled due to presence of DNA repair process in our system or in our body inside the cell. It will activate the survival response network to manage the stress response and cell control or regulate cell cycle checkpoints and increase the rate of DNA repair to survive the cells. If the damage it is excessive, irreparable, that will activate the apoptotic pathway and that lead to the cell death, or that mutation, it will be replicated and it will produce malignant transformation. So based on that, the literature classified the mutation to different classes. But before that, we need to understand or to identify what is the mutation. The mutation specifically, it is a change in the nucleotide sequence encoding portion of the DNA, which may alter the amino acid sequence of protein, or it is a change in non-coding region of the DNA, which has a potential for changing expression of the gene, for example, if there is a damage in the promoter region. There are many mutations which are classified to chromosomal or DNA-based. The mutation can also be categorized on the base of the function. The loss of function mutation cause decrease or loss of the gene product or the activity of the gene product, or can be the gain of the function mutation that cause increase in the amount of gene product or its activity, and sometimes create a new property leading to the toxic product responsible for pathological effect. These mutations, they call it point mutation, that affect on the protein structure and function, which is a chemical change in just one or few base pair in single gene. If the point mutation occur in gamete or in a cell that give rise to gametes, it may be transmitted to offspring. The mutation has an adverse effect on the phenotype of the human, and also the mutant condition is referred to the genetic disorder or hereditary disorder or the hereditary disease. Again, they classified these point mutation based on the kind of change. It is either base pair substitution or base pair insertion or deletion. So what is the pair-base substitution? It is the of one nucleotide partner in the complementary DNA and with another pair of nucleotides. It's either be missense mutation. If it is missense mutation, it will alter the codon, which still codes for amino acid, and thus makes sense, although it's not necessarily the right sense. Those cause a readily detectable change in a protein. The alteration in the single amino acid in the crucial area of the protein in the active site of the enzyme significantly alter the protein activity. Much often such mutations are detrimental, create useless or less active protein that impair cellular function. For example, mutation of single base pair in the gene that codes for one of the polypeptide of hemoglobin. The change of the single nucleotide in the DNA's template strand lead to the production of an abnormal protein. The second class of point mutation, we call it silent mutation. The silent mutation, they have no effect on the encoded protein, but the change in the base pair may transform one codon into another that is translated into the same amino acid. For example, if CCG mutated to CCA, the messenger RNA codon that used to be GGC would become GGU, and the glycine would still be inserted at the proper location in the protein. Other changes of a single nucleotide pair may switch an amino acid but have a little effect on the protein. The new amino acid may have properties similar to those of the amino acid it replaces, or it might be or it might be in a region of protein where the exact sequence of amino acid is not essential to the protein's function. موجودين معي النت كويس؟ سامعيني كويس؟
يا موجودة.
تمام. Nonsense mutation. The third class of point mutation, it is altering of codon to change an amino codon to the stop codon that lead to translation will be shorter than polypeptide encoded by the normal gene. Occasionally, such a mutation lead to improve a protein or one with novel capabilities that enhance the success of the mutant organism. The second class of point mutation, it might be insertions or deletions. Insertions and deletions are additions or losses of one or more nucleotide pairs in a gene. These mutations have a deleterious effect on the result protein more than substitution do. Messenger RNA is read as a series of nucleotides may alter the reading frame, which is from the separate grouping of the genetic message, and that make it the frame shift, or we can call it frame shift mutation. All the nucleotides that are downstream of the deletion or insertion, it will improperly grouped into codon, and the result will be extensive missense ending sooner or later in nonsense premature termination. If the frame shift is very near to the end of the gene, it will produce proteins that is almost certain to be non-functional. You can imagine what kind of frame shift will make the detrimental change if you look for your right-hand side and this diagram which represents the frame shift causing immediate nonsense. So the ability to diagnose a human disease at the DNA level has become possible because of rapid development in the DNA technology. The approaches that have been used for identify this mutation we mentioned earlier to classify it based on two subclass. Either it's called known mutation or we call it unknown mutation. Today, inshallah, we will focus on known mutations detection. There is a different of technology, DNA technology, we can detect these mutations. The gold standard technique to detect the known mutation, as you know, most of you know that it is polymerase chain reaction, or as we call it, PCR. It is a technique by which any piece of DNA can be quickly amplified without using cells in vitro. It was first reported in mid-1980 and had found numerous applications in both basic and research. It also allows numerous assays that depends on a quantitation of DNA or PCR, a popular technique with wide range of applications that depends on essentially three major advantages. It is a rapid technique, extremely sensitive, and robust. Its sensitivity allows the amplification of minute amounts of target DNA, even from the single cells. And various application in diagnosis, research, and forensic sense. Also, its robustness means that it is often possible to amplify DNA from tissue or cell that are badly degraded or embedded in some medium. This is the Pcr block, as you can see in this image. This is frequently, you can see it in most of the molecular genetic labs. They use the conventional PCR, or as we call it, thermocycler block. The PCR reaction, it has to have an essential component to set up. The first component it should be available is a template. The template, as we call it, or the sample of the nucleic acid that contain the desired target of sequence, which is the patient sample. The second thing is the primer pairs. Primers are short oligonucleotides. Usually it is between 20 to 40 oligonucleotides, but minimum it is 14 in length, 18 base pair in length, which complement to the target sequence and bind to the single-strand DNA. DNA polymerase, which the enzyme is required to amplify the target sequence. It's the thermostable enzyme that synthesizes copies of DNA with very consecutive cycle of PCR. DNTPs, or deoxynucleotide triphosphates, known as DNTP, which is one of four, either T, C, G. It is used in, it's become in concentration between 20 to 200 micromolar. The ideal concentration to be used in the reaction 50 micromolar for each is ideal concentration required. For each, the DNTP should be included in the reaction. Excess DNTP, it will inhibit the polymerase activity, DNA polymerase activity, whereas low DNTP concentration enhance the fidelity of the polymerase, but reduce the amplicon yield. The buffer is the PCR reaction service to facilitate the amplification by stabilizing the polymerase. Different PCR buffers are currently used, but the preferred one is Tris HCl, which must maintain pH of 8.4 at room temperature. The last important component in the PCR reaction is magnesium. It is an important cofactor for DNA polymerase. It is assist phosphodiester bond formation, and it is required for successful PCR amplification. So now the important thing to know, or the major part of the PCR to determine the primer to amplify the target sequence. To design our primer, we should generally have the following properties. As we mentioned earlier, the main of the primers, it must be correspond to the sequence on the template molecule. The length, it's between 18 to 24. Others, they prefer to be up to 40 bases length. The GC content between 40 to 60%, and start and end within 1 to 2 GC pairs. Melting temperature between 50 to 60 degree. Primer pairs should have a melting temperature within 5 degrees of each other, and the primer pairs should not have complementary regions. So practically, when we will want to design a primer, we can go to the website. There is a many websites online, which is free to design your primers. One of the very good application to design our primers is a Primer Plus. Also, we can use a Primer 3. These links, I place it in this slide, so you can practice by yourself. I will mention the whole step to primer design, then you can practice, but please consider this step. It is very critical in molecular genetics, not only in PCR. If you design your primers correctly, you will get a good amplification and successful target detection. So first of all, you need to open the NCPI on gene gate. Our target gene today, it's because we are looking for diagnosis of acute myeloid leukemia. We select FLT3 gene, which is one of the causal gene if it is a mutated lead to acute myeloid leukemia. So just type in the research field here the name of your gene, required gene, confirm it is placed on the gene gate. That will open this window for you by the name and abbreviation of your gene, and immediately you can select the Homo sapien version of your target gene. The next step, go to with the same page. When you scroll down, you will find in the navigation on your hand right side, select the reference sequence gene. That will open this page for you, represent the whole sequence, including the exons, introns area of your target gene. Then please press on the top of in the top of your page on FASTA. That will open you, transfer you to the nucleotide gate, which give you more details about your target gene. Click on the FASTA form of your sequence, target gene sequence, that will open you with this window, which consists, or it has the whole target gene sequence in FASTA form. The most important step here, you have to highlight the sequence feature. The request of highlight sequence feature, it will give you the specific coding region only, especially when you put your setting on the exon region. So you will show, after you change the setting to the exon region, it will give you the highlighted region. These highlighted region represent only the exons of the gene, of your target gene. Exons of your target genes are highlighted by the software, okay? After you highlight, select this, you have to know what is your target exons that include the mutation you are looking for, or you want to study or to investigate, either it is present in your patient sample or not. So from the literature, we know the FLT3 mutation. There is a mutation called in the ITD, or internal tandem duplication, located in exon 14. So I moved till found the page number 14, which corresponding to the exon 14, and which is highlighted in front as shown in front of you. So this is exon 14. Then at the bottom of this, the software, you can click on the FASTA sequence. It will move you to this only the only sequence of exon 14 that include known mutation of internal tandem duplication of FLTG. So the next step here is crucial. Our target here to amplify or to detect or looking for the mutation in this exon, which is exon 14. And this sequence present in front of us, it's exactly the sequence of the exon from C to G at the 3' end of the sequence. But during the amplification, as you know, if you are worked previously with the Pcr, it will never ever show you any amplification from the first base during the reaction. So what we need to know at this stage, this happened, become before the exon region started. Hello, are you with me? النت كويس؟ تسمعوني كويس؟ Please let me know if it is disconnected.
لا سحر مو واضح. سحر. سحر معانا؟
واضح السكرين؟
لا، شكله فصل.
طيب واضح السكرين الآن؟
واضح.
تمام. المعذرة على الاتصال، بس معليش دقيقة عشان طيب. So we will change the length of the sequence. If you look for navigation panel on your right hand side, you will find the length of the exon 14. It is start from 71,355 till 71,848. As we mentioned before, we are not sure the amplification will start from the first base of the exon. We will take part of the intronic region before and after exon 14. So we will add about 200 bases at 5' end. So it will become 1755. I will just add more of intronic region before the exon 14, and I will add 100 bases at 3' end after the end of exon 14. And that will become, it will change from 71,487 to 587. So after that, we will copy the sequence of exon in Word file. The yellow highlighted, this is represent only exon 14 sequence. However, the bottom, it will represent the highlighted, started from the yellow till the last of yellow represent the exonic region. The unhighlighted bases before represent the 200 bases extra of intronic region and 100 bases extra after exon 40, which is that this will be the target sequence to find a complementary primer to amplify the sequence. So we'll copy the sequence with the exon 14 within the intronic region, paste it in a primer blast software. And we need to determine at this stage the length of required forward primer and a reverse primer that we want to design. And we don't need to change anything, the setting of the primers parameters regarding the target, the PCR product size, that melting temperature is suitable for us, so we will not change anything at this stage. Here, how can I determine the beginning of the primers and the end of the forward primer, we will back to the word. Okay, we will highlight the first 10 bases of the intronic region. Okay, we will look for it. Where is it placed? All right, and the end of the exonic region of exon 14. So this will give us the length of the first forward primers from 1 to 63. Exactly the same step, we will do it to determine the length of the reverse primers, because I did not mention it here in my slides, but it is exactly the same. We will take the last 10 bases of the intronic region before after the exon 14 to be selected and determine the reverse primer length. So then we will click on get primers. That's this window. It will be show us the possibility of all the primers can be used to start our Pcr reaction. The software gives us about 10 possible pairs of the primers. The first one, which seems suitable for us, its length, it will give us a product length 418. It shows us the sequence of forward primers, the reverse one. When I copied the forward, pasted on my sequence of which is on the Word file, it show me where is the beginning, which is the pink colored primer of my sequence here. This is the forward primer, and the second reverse primer, which is the sequence. It is also placed, and I confirmed that will flank my target sequence, which is exon 14. It will be amplified as required. After that, we can copy the sequence, go to one of the companies which is responsible to form these primers. We can order it online, open the custom standard DNA OLEX, enter our sequence of primers, determine all required information, pay for this primer, and this will maybe take 1 week or more or less, and they will send it for you. I think this facility available in the UK very easily. Next, we will go, so after our Pcr reaction primers are ready, we will go to set up our Pcr reaction. The most important thing before go to amplify our target sequence, we need to make something we call it master mix. The master mix, we make it to amplify our target gene, which is FLT3 in this condition, or in this case, at ITD mutation. We have component, we have to prepare our Pcr component, which is Pcr buffer, DNTB, our primers, reverse and forward. By the way, the primers become from the company, it is highly concentrated. It's to 400 picamole. We need to dilute this primers to the required concentration according to the insert sheet for amplification for the target gene. For example, here we just need 10 picamole of the forward and reverse primers of FLT3. Also, we need DNA polymerase. In this reaction, we use tag polymerase enzyme. and also the water to facilitate the reaction. So the final concentration, it is highlighted here in the reaction setup. Each volumes are set in front of you before you start the reaction. And the last column here, it's important to make the master mix based on the quantity or how many samples do you have. So when I prepare the master mix for my reaction, I need to find how many case or how many patient or how many sample or cell line in case of your PhD researcher I have to prepare sufficient master mix according to the manufacturer instructions. We need 48 microliter for each sample to amplify the target sequence. Okay, so this 48 for each, and for in our case, for example, we have 4 patients or 4 cases plus 5 3 patients. We have positive control. It must be included in our reaction, which help guide us to compare between our patient and the control positive. And the last thing here, which is NTC. NTC represent non-template control. That means here in this reaction, instead of put a DNA, I will put water. This NTC control, it is very important to confirm the clearance of my reaction. I shouldn't have any amplification in case I have water instead of DNA. If I have amplification in this sample or in this lane, that means I have contamination during my PCR and all my results are not specific, are not correct, and I should terminate this experiment. Okay, so and after, for example, here, plus we have positive control NTC, we need to add 1 extra sample just in case we lost some of quantities of the master mix during the pipetting in my experiment. So I need to prepare master mix for 6 samples, not for 5 samples, 1 extra. Then I will multiply that 6 sample by the required volume of the component. For example, if I have 6 samples and the required volume for each sample is 5 of buffer, 5 times 6, it will be 30. 6 times 4, it will be 24. 6 plus 2, 12, and etc. The total, it should be sufficient for 5 samples of master mix plus 1 extra. Okay, so in the PCR tube, we will add 48 of the master mix plus 2 of microliter of my patient DNA. Okay, so the total reaction, it will be 50. Once we prepare our PCR reaction, our reaction tube, each tube, it has patient DNA, master mix, and for the first 3 patients. The 4th, which is positive control, I should have positive, non-positive for FLT, ITD, mutation, and the 5th tube, it is 2 microliter of water plus 48 of the master mix. We will put this in PCR tube reaction, mix it, then transfer my tubes to the Pcr block. In the Pcr block or thermocycler, the polymerase chain reaction, it will be start. Depends on a sequence, it will act the primers for a new DNA synthesis at defined target sequence. The idea is to selectively replicate only the target sequence. So why we need the primer with a specific region at a specific, it would include a target mutation to be amplified or to investigate or to detect, because I don't need the whole genes in the DNA of the patient sample. As we know, we have more than 22,000 genes in our DNA. But here I'm looking for a specific gene, which is FLT3, and I'm looking specific area of FLT3, which is exon 14. And within this exon 14, I expect one mutation point, a mutation should be amplified. Okay, so that's why I do PCR with specified primers to amplify only the target sequence of the target gene. Okay, so the reaction, it will be consist of series of cycle of 3 successive reaction conducted at different temperature. This is processed by thermocycler or Pcr block. The first, there is, as you know, there is 3 phases of the Pcr reaction. The first called denaturation, the second is annealing, and the third is extension. So let's understand what happened during the denaturation step of the Pcr. The denaturation of the Pcr, it is to separate the double strand DNA into 2 single strands by breaking the hydrogen bond between the nucleotide base pair. The initial denaturation step is needed at beginning of Pcr before the cycling of 3 stages begin. The initial denaturation step usually occur at around 94 degree or the range of 94 till 98 degree up to 2 minutes. A higher GC content require a higher and longer denaturation temperature. The denaturation step is usually set at a sample temperature as initial denaturation about 94 degree, and it will require only from 10 to 60 seconds. And it is better to avoid extend the incubation at this denaturation temperature to prevent inactivation of the polymerase or damage the DNA templates. Okay, if we have a newly designed primer, just like in our case of FLT3, we just receive it from the company, we need to do the optimization step before to start the actual Pcr reaction. We need to start to study the amplification of the annealing temperature of the primer at a specific stage. We will discuss this in the next slide. The second step of the Pcr reaction is annealing. The primer anneal or bind at the complementary sequence of the denatured DNA strand. It will only occur successfully if temperature and the primer pairs have been optimized. As I mentioned, we need to do optimization for our newly received primers before do the actual. So for example, if we request the annealing temperature for primers to bind with the target gene between 52 and 58, we need to select 3 temperatures of annealing temperature set up in the Pcr thermocycler and start to our Pcr reaction and look for the best annealing result or binding between the primer and target. How can I confirm this is the best, or how can I stabilize this temperature, it is the best temperature to amplify my target sequence by look for the thickness of the band on a gel electrophoresis. So for example, let's say our ideal annealing temperature for FLT3 ITD, it is 56 degree, and the annealing time is maintained from 15 to 60 seconds with optimum time at 30 seconds. If the annealing temperature is too high, the primer do not anneal efficiently. And if it is too low, the primer may be bind non-specifically to the template. The third phase of the PCR reaction is the extension. At this stage, or the final stage of the cycle, that occurs at the temperature that allow optimal polymerase activity of binding the nucleotide to the annealed primer, resulting in exponential amplification of the template strand. Usually, the extension step can be carried out at between 70 to 80 degree for 1 to 2 minutes. Extension temperature and time are specific to the polymerase. For example, we have TAG operates best at from 70 to 80 degree for 1 minute for the first 2 kb, and it requires another minute per additional kb. For example, PFU as enzyme, meanwhile, it is function at 75 degree and it requires 2 minutes per 1 kb or kilobase of the DNA. Overextend this phase will create higher error rates. Annealing and extension can be compressed into the one stage if the corresponding temperature are similar. And this is referred to as two-step PCR because the 3 stages have been reduced to 2. Two-step PCR is most often used when the melting temperature of primers are within the normal range for extension activity. One final extension period of 5 minutes is performed at the end of thermocycler, which results in addition of adenine to the 3' end of amplicans. This residue is needed for different downstream application, including cloning. So when will we take a PCR overview about the PCR? It permits selective amplification. Some information about the sequence of the target DNA is required. The information is used to design 2 oligonucleotide primers, often about 18 to 25 nucleotides long, that are specific for 2 sequences immediately flanking the target sequence. The primer must have sequence that have a very high degree of base complementary to the sequence flanking the target DNA. Most of PCR reaction is to amplify single DNA sequence. So it is important to reduce chance that the primers will bind to other locations in DNA than desired ones. It is important to avoid repetitive DNA sequence too. To avoid binding of 2 primers to each other, the 3' sequence of 1 primer should be complementary in sequence to any region of other primers in the same reaction. So this should be considered at the step of a primer design to ensure that primers doesn't undergo internal base pairing as well. The primer sequence must not include inverted repeat or any self-complementary sequence more than 3 base pair in length. The temperature for a primer annealing step should be set as high as possible while still allowing base pair between the primer and its binding, its melting temperature, and that melting temperature and primer annealing temperature depends on the base composition. Optimal uses primers with GC content between 40 and 60. Primer design is aided by various commercial software programs as we used in earlier slides. There is one step I did not mention it in my slides. If you after you design your primers and you selected from the software, you can go to the another software that make like test for your primers. Is it pass these criteria or not? Is it all successful? All the considered criteria of a primer, good primers, is matched or not? I forget actually to write it down, but if you are interested, I can mention it later, inshallah, in part 2, to check your primer before you send it to the company to purchase it. After we amplify our target, still we need to visualize this amplicon. The result amplicon can be visualized through staining with dye or through labeling with the fluorescence nucleotide or primers. If we are working on conventional PCR, we will go to the electrophoresis reaction and we will use dye to detect the amplicons which or a PCR product which might amplify our target sequence which containing the mutation. The agarose gel and visualized molecule demonstrate the size and the quantity of the DNA product in comparison to DNA ladder. The ladder, which is a product, you can purchase it from the company. It is consists of the fragments of DNA with different lengths. These lengths, it is like for us a guider to determine our target size based on the known size of DNA ladders appear, as you can see in this image on your right-hand side. Also, preparing the gel, it should be under specific, we have many considerations to prepare the gel. The most important thing to prepare the gel at a step of the visualization of the target sequence, we need to select a specific concentration of the gel suitable to detect our target gene. For example, if we have a short or a molecular weight, a small molecular weight of the target sequence or the target gene, we need to have a condensed or highly concentrated gel to catch my target sequence. If I have large molecular weight, I need to prepare slightly diluted gel to apply the gene and enable it to transfer through the gel during the electrophoresis. There is specific consideration. I did not mention it, but again, if you are interested, I can provide you a specific or direct guide based on the molecular size, molecular weight of your target sequence. The electrical fields cause DNA to migrate through a gel matrix with differential speed. They can be tagged with other molecules for easy identification through color or fluorescence. Shorter fragments move at more rapid pace, so the approximate size can be identified by the comparison between the PCR fragment and the DNA ladder to confirm the consistency. So finally, our patient who's Noura, it is in lane, we place it in lane 4. If you find the lane on the gel after the electrophoresis is done, we will take the gel, place it in under the camera. We will switch on the UV light, and it will show in front of us exactly the same image in front of you. This is represent the gel, and these bands represent a targeted, successfully amplified sequence for FLT3 gene. How can I discriminate, or how can I distinguish, between the target sequence, this is normal or mutated? As I know from the literature, the ITD mutation, it is a duplication or increase in the size of the target juxtamycin, which is the receptor of FLT3. It is internal tandem duplication. So it is increase the size. What do I expect to see on the gel? Increasing the size, that means I have another band which represent increase or extra size for the target gene. So the first patient in lane 2, it doesn't have another band, which means this patient doesn't have the mutation of FLT3 ITD. The second patient in lane 3, again, this is normal patient. It's amplified the target gene, but it doesn't find any mutated cell. It has internal tandem duplication. Nora's sample, it was in the 4th lane. She has a positive ITD FLT3 mutation. How can I know that? Present of the extra band, which represent extra size of the FLT3 gene. When I compare it with the positive control, which I know earlier, this is positive patient, and the extra, it doesn't have a specific length or a specific size. Extra or the duplication, it's either from 3 base pair extra till 300 base excess or duplication. So if you look for your positive patient, it has a second band which represents a cancer cells that has a mutation of ITD. Then the second band which represent the normal DNA, which is within the normal cells, it doesn't have a mutation. So patient in lane 5 and 6, again, those are negative of FLT3 ITD. If you look for the lane 8 here, it doesn't have any bands which represent the NTC reaction. If you remember, one of our reaction, we add only water instead of the DNA, which that means the primers, it doesn't find any target to bind with. So nothing happened, no reaction, no amplification, so no band on the gel. That means this reaction, our PCR amplification, it is a clear, successful, and all these bands we can see in the front, it is correct. There is no incorrect or non-specific product has been detected, because my NTC, it is clear, there is no contamination in my reaction. So the final molecular genetic report for Noura was diagnosed with AML. And the AML prognosis is based on specific CBC result. And also we looked for markers on leukemia cells by flow cytometry, the chromosome translocation in cytogenetic, and the abnormalities found in the gene mutations of FLT3, which is positive. and that confirmed this condition. It is AML positive, and the doctor start the treatment plan with Noor. So the next question is all point mutation we can detect it by the same PCR technique? The answer is not. There is wide variety of PCR approaches that have been developed for specific applications. The variety of method for detection of point mutation as well as, so each specific mutation or kind of mutation, it has different type of technique or molecular genetic technique to detect this mutation. Not all point mutation we can detect it by direct PCR. Not all the mutation we can detect it by as FLT ITD3. So usually when we start to detect or investigate for a specific mutation, we should ask ourselves 10 questions. What type? The first is what type of nucleic acid going to be analyzed? Is it DNA or RNA? What kind of specimen I'm going to be analyzed? Is it blood? Is it bone marrow? Is it tissue? It is FNA? It is amniotic fluid? What kind of samples I have? What's the mutation? What type of mutation? As earlier we mentioned, there is a variety of point mutation. We have deletions, insertions, we have base substitutions. I have a deletion of only one base pair. I have insertion of 3 base pair. I have 3 substitution of 1 base pair. I have substitution of 10 base pair. I need to know what kind of mutations I'm looking for and how large this number of potential mutation to be detected. I need to know the potential, the ratio between the wild type and the mutant's allele. I need to know what's how reliable this method I have to select to investigate my mutation. How does this test it will perform? Is it it is routine, or it is something very rare? This rare mutation, I need a very specific technique to design to detect this mutation. What kind of quality assessment can be achieved? All these questions I should be put it in front of my table to select a best and appropriate choice of method to find or to detect the target mutation. As we mentioned, today we will focus on known mutation, and this is give us an advantage to go through which PCR technique or which molecular technique to select to find this mutation. I have a couple of molecular technique to select based on my mutation criteria. One of the frequently technique has been used in molecular lab is restriction fragment analysis detect restriction to detect restriction fragment length polymorphism, or as we know it, Rflp. This indirectly detects certain differences in nucleotide sequence of DNA molecule, and I need gel electrophoresis to sort the DNA fragments that treated restriction enzyme by the size, digestion of the DNA sequence by the same restriction enzyme may produce different band pattern if the restriction site affected by the mutation. As an example, the sickle cell anemia, it is happened because there is substitution of of the base A to T that will lead to destroy a restriction site for digestion on that used by MST to digestion or restriction enzyme. The wild type gene contains a 3 cleavage site for the enzyme MST2. I have this target sequence of beta-globin gene. It has a 3 cleavage site or restriction site for MST3. As we know, the base substitution from A to T that destroy the middle position of the MST2 restriction site that affect this site. And after the restriction or incubation of my target sequence with MST2, the PCR product, if it is not mutated, it will produce two band visible by electrophoresis. One length, it will be 1.1 kilobase, and the second 0.2 kilobase. If this position it's not destroyed by the mutation, I will see in the electrophoresis two bands. Okay, if it is destroyed by the mutation on the beta globin gene, I will have only in electrophoresis one length or one band, which is 1.3 base, that represent homozygous mutation. To clear it, we have this gel, and I have a family, the mother, father, and the child. And also I have my control for normal restriction digestion. If the individual has heterozygous, both mutant and wild type copy of the gene, okay, I will see the three band will be visible in the gel. Like in the father condition here, he has three bands. One is 0.2, the second 1.1, and the third 1.3. That means part of cells has a mutated gene, and the second part of the cells with the extracted DNA, it is clear. Normally, it doesn't have any mutation. So these two fragments, 1.1 and 0.2, represent the normal genotype of the beta globin gene, and the 1.3 band represent the destroyed restriction site, and that show me the length, the whole length of the target sequence, which is the 1.3. Similarly, the mother, if we look for the electrophoresis, it has a heterozygous genotype of A to T base substitution for beta globin. So mother and father, they are a carrier for sickle cell anemia. However, when we look for the child, he has only one band which represent 1.3 kilobase. That means he has a homozygous mutation and he is a patient sickle cell anemia. When we compare it to our normal control, there is no destroy in the restriction site, and I have two bands in the normal control. So in each technique, there is limitation. For Rflp, by simple Rflp, one mutant cell may be detected in 50 to 100 non-mutant cells. The detection limit can be lowered significantly by application of the mutant-enriched PCR. So the usually detection of the Rflp, it is between 50 to 100 cells. More than we can't catch this mutations. They make a modification for this technique. They call it a mutant-enriched PCR. The mutant-enriched PCR is the important modification for this technique. In the original report, the artificial or primer-mediated restriction site were introduced in wild-type DNA by application of mismatch primers localized in the direct vicinity of the possible site of a mutation. As a result of base substitution, uncleavable restriction site are generated from a mutant, but not from the wild type alleles. Subsequent to the first round of the applications or amplification, wild type DNA is eliminated by a restriction digest. In the second round of amplification, applying the same primer or hemi-nested primers, ideally BCR product representing the mutant allele, because we amplified now the whole mutant. So most of the cells are, or most of the templates are mutated. The only will be amplified that the mutant-enriched PCR would also visible with natural restriction site if present. So if it is present, you will find clearly there is no amplification or cutting. If it is not, you will find the fragmented DNA after the digestion by the target restriction enzyme. The quality assessment for Rflp are analyzed by gel electrophoresis and ethidium bromide staining. Other method of detection we can use hybridization or mobilization, but this is less common. The specificity of this technique 100% is achieved when appropriate restriction enzyme are used. As a quality control, different allele, variant or wild type, and a mutant DNA must be included in each analysis. We should include these controls to facilitate our data interpretation. The recognition sequence may be destroyed by errors of the tag polymerase enzyme, so we should be careful or select our enzyme. In general, errors due to misincorporation will become detectable only when we have a high number of PCR cycles and or sensitive detection method are used. In the mutant-enriched PCR, false positive result will be obtained when critical number of cycles is exceeded the second PCR subsequent to the restriction. The second application for a specific PCR, we call it allele-specific oligonucleotides. This is when this is modified kind of the PCR to detect mutations, but in different way. For example, the principle of the ASO hybridization, it is designed, the DNA chip is based. This is frequently used in this application. And this method for DNA product generated by PCR, the detection here is the different from the conventional PCR. In the conventional PCR and also Rflp, the detection was on the gel, but here we will plot our PCR product and duplicate a membrane. One membrane is dotted or hybridized with an oligonucleotide correspond to the wild type sequence and other to the mutant sequence. Normal individual, it will show hybridization only with the normal probe. The homozygous individuals show hybridization only with a mutant probe. If heterozygous individual hybridized with both probe normal and a wild type. So this is the membrane. When we make the PCR product, we'll now apply this PCR product in this membrane. The probes of normal or wild type is fixed on the membrane, and the mutant one is also fixed on the membrane. After the PCR or the amplification step, we will apply our samples on both membranes, the mutant and the normal and the mutant. So this, for the genotyping for the patient number 1 here, this is, we have only the amplification or hybridization in the wild type. So this patient, it is, he has a wild type gene of target, wild type amplification of the target gene. And the second patient, he has a hybridization on the mutated prop. That means this patient homozygous mutation detected of the target gene. The third patient, he has hybridization in both mutated and a normal prop. That means this patient has heterozygous mutation of the target sequence and so on. We can use 32P labeled prop for the detection also, or non-radioactive detection in direct or reverse dot plot. We can use evidine pyridoxidase conjugate can also be applied to detect the mutation. The third technique for detect known mutation, we call it RT-PCR or reverse transcriptase PCR. The discovery of retroviral reverse transcriptase in early 1970 ultimately made RT-PCR possible. When I can apply or select this technique, if I have RNA sample, if I would like to detect a mutation in RNA or at a level of messenger RNA, I can use RT-PCR or RT-PCR. It will enable us to detect the mutation at the level of RNA. So this is advantage of this technique. The template is total RNA or purified poly-A tail messenger RNA and initial reverse transcriptase step to produce the end product within known as complementary DNA, as we call it. We will produce cDNA. How can I form cDNA? This is a process. We will discuss it either today or next time. The cDNA is not subject to RNA's degradation, making it more stable than RNA. So the target sequence here, it is RNA. But the problem is the RNA sequence, it is not stable. So I need this technique to detect the mutation at the level of messenger RNA, but on the form more stable. So we will return the RNA to the DNA. So we call it complementary. So we will build DNA again, but it is complementary and it's corresponding to our RNA sequence. The cDNA serves later as a template for exponential amplification using the PCR. The PCR or RT-PCR can be undertaken in 1 or 2 steps. One step RT-PCR combines the RT reaction and PCR reaction in the same tube. Only sequence-specific primers may be used. Or in 2 steps RT-PCR, that synthesized cDNA is transferred into the second tube for PCR. The oligo or random hexamer or gene-specific primers can be used. The primers are generally preferred as they hybridized to the 3' poly-A tail in messenger RNA, whereas the random primers prime anything including ribosomal RNA. RT-PCR requires an RNA template, enzyme, nucleotide, buffer, and thermocycler to produce the RT-PCR product. It is also used in research laboratories to study the gene expression, for example, in experiments to distinguish exons from the introns, and can be used clinically to diagnose genetic disease and monitor drug therapy. So this is generally the step for RT-PCR. RNA consists of start, codon, We will use oligo primers. Also, the reverse transcriptase enzyme, it will be used in the reaction. After that, the primer, it will bind in target region. The reverse transcriptase, it will bind to the end of the primers, and the extension, it will be start to make or form cDNA, or the complementary DNA. The fourth technique to detect point mutation is the PCR specified or designed to detect small indels or small deletions, such as a deletion of most known or frequent happening, the deletion in CFTR gene at position 508. This mutation, it is due to the deletion of 3 base pair, which is less than 20 deletion in codon 508, and that result of loss of phenylalanine residues at amino acid position 508. That will cause cystic fibrosis disease. The PCR amplification of 89 base pair region spanning the site of the deletion in polyacrylamide gel electrophoresis, and the allele migrates faster than wild type. Heterozygous subjects were positive for both normal and the mutation gene fragments. However, the homozygous abnormal subjects were positive for mutation gene fragment and negative for normal gene fragments. So the whole idea about detection of small indels or deletions of target sequence or a specific gene is designing the primers. Here, the gold standard or the major player, we design a primer that cover the missed or deleted region of the target sequence. So we can understand from the gel electrophoresis, if there is no amplification, that means the patient, he doesn't have a deletion. If there is bands or amplification at different places, that means this patient heterozygous for this mutation, he has part of the DNA with a deleted CFTR, and part of the population, it has no deletion. If there is both of the or the patient has homozygous deletion of the CFTR gene, we will find the amplification, the band, it will be appear in the gel. If we have a larger deletion, we can move on to multiplex Pcr. We can use more than a primer at the same reaction. As an example, if we have a larger deletion like 300 base pair deletion, that also corresponding to the insertion allele as well. So we have to design primers that cover the region missed from the original gene, and that region should be known so we can detect the missed mutation or missed sequence of the target gene on gel electrophoresis. This is what we call it gap Pcr. Gap Pcr, that specific primers are designed to flank known deletion. The principle of gap Pcr is based upon the inability of a primer to generate Pcr product unless a deletion join flanking sequence together. If a deletion is present, the Pcr amplification will occur and the product is examined by the electrophoresis. The illustration to show you how can we detect the mutations by design primers that cover a flanking region with missed sequence, and generally classification of the mutation can be determined, or genotype of the mutations can be determined on gel electrophoresis. If we have massive deletion or gross deletion or insertion, we will go to another or a more advanced technique to catch or to detect these mutations, because we never ever can design primers, multiplex primers with high specificity to catch missed sequence of the target gene. We need to go further for more advanced technique like MPL, or multiplex ligation dependent probe amplification, or Southern plotting technique. In MILPA, or multiplex ligation dependent probe, it is commonly applied to screen deletion and duplication of up to 50 different genome, DNA or RNA. So this is an advantage, this technique, we can use it for DNA and RNA. Altogether, gene deletions and duplications account up to 10%, and in many disorders, up to 30% of disease-causing mutation. In this technique, the probe set is hybridized to genomic DNA in solution. Each probe consists of two halves. One half is composed of target-specific sequence and universal primer sequence, and the other half has other more sequences, variable length random fragments to provide the size differences for electrophoretic resolution. A pair of probe is hybridized on the target region so that they can then be joined by the use of ligase. Contiguous probe can be amplified by Pcr. After Pcr amplification, the copy number of target sequence, for example, deletion or duplication of the target sequence, can be determined and quantified using the relative peaks heights. So we have the panel of pairs of the DNA probe hybridized to the multiple sites within the target sequence. The following successful hybridization of each probe is ligated by ligase. So the ligase, it will not bind till the successful binding of the target site it happen, that will show us the successful hybridization. Each pair of a probe, it should be include hybridization sequence, which is clearly illustrated here. A Pcr primer sequence, which is the orange sequence or piece, and the same of the all probe pairs, and the stuffer sequence, which is different in length for each probe. So the MILPA probes, it should be designed at the specific way to detect the mutations or a duplication by this technique. The hybridized and ligated probe only can be amplified by the Pcr using one primer pair. The amplimers are unique size, depends on the size of the stuffer sequence. Amplimers are resolved by capillary electrophoresis. Currently we work on the capillary electrophoresis on genetic analyzer instrument. We will go for this technique, inshallah, in the section of DNA sequencing. Relative amount of the probe amplification product compared to the control DNA sample that reflect the relative copy number of the target sequence. So how can I find the result? It will be appear in front of me as peaks. These peaks represent duplication or a deletion based on heights. So the result or MALPA analysis, for example, let's take a patient, he has a DMD, which is X-linked recessive disease, expressed as a dosage or the ratio of exonic peak reference peaks is the patient with exonic peak compared with the healthy controls. So in the first condition, the male, he has 5 exons deleted. There is no amplification at all in this area, and this is represent a deletion of this patient. The second female, which is a carrier for DMD, she has a partially delete of the target exons, which is a carrier for the deletion. So the dosage, it's represent only one copy, which inherited from the parents, but there is no another copy. So it will be less than expected. The third condition or the third patient, he has 10 exon duplication. So this case, the length of the exons, it is greater than expected. So this patient, he has a duplication and easily can be detected and visualized on the software. The last technique we will talk about for detection a known mutation today is a real-time PCR. Real-time PCR or qPCR or quantitative PCR is a widely used method to quantitate DNA or RNA. RNA is copied by using the reverse transcription to make the complementary DNA strand. Amount of DNA is measured after each cycle via fluorescence dye that yield increasing the fluorescence signals in direct proportion to the number of PCR product molecules generated. Fluorescence reports used include double-strand DNA, binding dyes, or dye molecules attached to PCR primers or probes that hybridize with PCR product during amplification. The instrument generates an amplification plot that represent the accumulation of the product over the denaturation of the entire PCR reaction. As all the PCR reaction, we have a specific component to set up the reaction. In the real time, we need tag DNA polymerase, we need a reverse transcriptase, DNTP, magnesium concentration, it should be considered, template, which is the patient sample, and tagman assay product primers and probes. There is 2 kinds of detection technique usually or most frequently used in the labs to detect the target mutation. The first is a detection using the tagman probe. If it is a hydrolysis probe, which pair a reporter dye, often fluorescence, fluorescein or FAM at its 5' end, and the quencher known the most frequently TAMRA attached to the 3' end of the oligonucleotide. Under normal condition, the probe remain coiled on itself, bringing the fluorescence dye near the quencher, which inhibits or the quenches of the fluorescence signals of the dye. The oligonucleotide, if the tag polymerase has homologous region with the target gene, and thus when the target sequence is present in the mixture, it binds to the target or the sample DNA that will lead to break down this probe, and the fluorescence it will be released and detected by the detector. As the tag polymerase start the synthesis, sorry, the new DNA strand in the extension stage, it is called degradation of the probe by 5' end nuclease activity, and the fluorescein is separated from the quencher, as a result of which fluorescence signal is generated. In each cycle, the number of signal molecules increase, causing increase of the fluorescence, which positively related with amplification of the target. We can select another approach. Instead of using the tag probe, we can use cybergreen. The cybergreen, it is a dye. It's emit prominently fluorescent signal when it bind at the minor groove of double-stranded DNA. So any double-stranded DNA, it will the cybergreen, it will bind to it and release the fluorescence. We can use also the ethidium bromide or acridine orange can also used instead of cybergreen, which is better used for its higher signal intensity. The cybergreen is more preferred than the Tagman probe, as it can provide information about each cycle of amplification, as well as about the melting temperature, which is not obtained from the Tagman. So the detection approach here, difference between the difference between cybergreen and Tagman, it's the way of telling us is this the amplification taking place or not. The disadvantage of this approach, it is lacking of the specificity. As we said, any double strand, the cybergreen find it in the reaction, it will catch it, and fluorescence, it will be released. This double strand, it's maybe the primers, it's bind together, maybe it's not the specific target, maybe it is the target, we don't know. We can't confirm, we can't be certain this is specifically the target sequence or not. Real-time PCR can accomplished in 2 steps. And this the first step, CDA, we will start with formation of cDNA by RT-PCR. Then 10% of the cDNA is transferred to the separate tube for a real-time PCR. One step RT-PCR combines the first strand cDNA reaction and the reaction, the real-time PCR in the same tube, amplifying the reaction setup and reduce the possibility of contamination. Gene-specific primers are required because using oligos DT or random primers will generate non-specific products in the one-step procedure and reduce the amount of product of interest. For the result interpretation produced from real-time PCR, we should consider 4 important things that help us to analyze our data. Baseline, the threshold, the CT, and standard curve. The baseline, which refers to the signal level during the initial cycle of the PCR, usually cycle 3 to 15, in which there is a little change in the fluorescence signals. Low-level signal of the baseline can be equated to the background or the noise of the reaction. The baseline should be set carefully to allow accurate determination of the threshold cycle or CT. The baseline determination should be taken
سحر الصوت. سحر.
Hello.
أهلا.
Sorry for the inconvenience. I don't know that disconnected, the internet disconnected again. We are almost done for today. The last 3 or 4 slides to explain, which is very important. Can you see the screen now?
واضحة.
Yes. We stop at the threshold. The real-time PCR instrument software automatically set the threshold at 10 times the standard deviation of the fluorescence value of the baseline. CT value, it is a threshold cycle, is the cycle number in which fluorescence signals of the reaction cross the threshold. CT is used to calculate the initial DNA copy number, and it is inversely related to the starting amount of the target. A sample with a twice starting amount will yield CT 1 cycle earlier than sample that contained half as many copy of the target per year of the amplification. We will see all this information practically on the curve or in the data. The standard curve, it's diluted series of known template concentration used to establish the standard curve for determining the initial amount of the target template. The log for each known concentration in the dilution series in x-axis is plotted against the CT value for the concentration on y-axis. The concentration chosen for the standard curve should encompass the expected range of the target. So this is the amplification plot. This is the baseline we just explained. It is the noise. We can't consider this amplification or successful amplification until we will reach the threshold. After the threshold, the amplification will show consistency of curves that represent the amount of the target region, the massive or the high dose of the target sequence. It will appear earlier in earlier sequence when it is have loss concentration or low copies of the target sequence. It will appear later. The standard curve here, we have considered 3 major things to approve our result. Before to interpret our result, we need to look for standard curve first, to look for the efficiency of the PCR reaction. It should be 100%. That means the template doubles after each thermocycler during the exponential amplification. The slope of the log linear phase of the amplification, it should be accurate and reducible, show a reducible result. The reaction should have efficiency as close to 100% as possible, equivalent to the slope of minus 3.32. The good reaction should have an efficiency between the 90 to 110%, which correspond to the slope between 3.58 and minus 3.10. The third things we should consider the correlation coefficient, or R2. The correlation coefficient is a measure of how will the data fit the standard care. The R2 value reflects the linearity of the standard curve. Ideally, R2 is equal 1, although 0.99 is generally maximum value. So if we represent the standard curve with efficiency high 100% and or between 90 to 100%, the R2 it's equal to 1, that we can consider and move on to translate the result or interpret or analyze our target sequence by real-time PCR. The absolute quantification describe the sample of known quantity are seriously diluted and then amplified to generate standard curve. Unknown samples are then quantified by comparison with this curve. The second approach to interpret the result using the relative quantification, which is the mostly frequently approach used in labs, that describe expression of gene of interest on sample. For example, I have a treated sample. This patient have a cancer. He's started treated with a chemotherapy. We will compare it to the expression, the gene expression of the sample for a patient untreated. This result are expressed as a fold change, increase or decrease in expression of the treated in the relation to the untreated. The normalized gene, for example, in our case, for example, we'll talk about the beta-actin, is used as a control for experimental variability in this type of quantification. One also good data we can or analysis beneficial from real-time PCR, it's melting curve or dissociation curve. This is one of the applications of real-time PCR. We can use it for quantitative detection of the target mutation. We can use it for qualitative detection of target mutation, or by using a melting curve analysis. A melting curve chart is a change in fluorescence observed when double-strand DNA with incorporated dye molecule dissociates into the single-strand DNA as a temperature of the reaction is raised. For example, when double-strand DNA bound with cybergreen, one dye is heated, a sudden decrease in the fluorescence is detected when the melting point is reached due to the dissociation of the DNA strands and subsequent release of the dye. The melting temperature of a nucleic acid is affected by length, the GC content, and the presence of the base mismatch, among other factors. Different PCR products can often be distinguished by their melting characteristics. Probe-based technique is sensitive enough to detect signal nucleotide polymorphism, and we can distinguish between homozygous wild type, heterozygous, and homozygous mutant allele by dissociation pattern product. The whole earlier techniques we talked about is about detect one point mutation only. Then scientists generate approach, they call it multiplex real-time PCR. That means we can detect more than a mutation at the same reaction. We don't just set up the reaction to detect only one mutation, one deletion, one insertion. No, we can use more than one primers at the same reaction to detect more than a mutation at the same time. So the amplification and specific detection of two or more genetic sequence in the same reaction, the real-time PCR multiplexing may be used to produce a qualitative or quantitative result. The three benefits of multiplex: it's increase the throughput, reduce the sample usage, and reduce reagent usage as well. Multi-dye real-time PCR fluorescence chemistry is being used. Each genetic sequence will be required as a different reporter dye. The advantage of the real-time PCR include ability to monitor the progress of the PCR reaction as it occurs in real time. It is enable us to precisely measure the amount of amplicon at each cycle, as we mentioned. This is the advantage of cyber-green approach, which allows slightly or highly accurate amplification or quantification of the amount of starting material in the sample. It is increased dynamic range of detection, and also the advantage of real-time is amplification can be detected occur in single tube or will eliminate post-PCR manipulation. As we mentioned also, real-time has different applications. We can use it for gene expression profiling that assess a relative abundance of transcript to determine the gene expression pattern. And this is, we will mention it again at in part 2, inshallah. We will talk about the functional analysis functional analysis or techniques to detect that mutation. The second application is viral titer determination assay to quantify the viral copy number in samples, and this is often accomplished by comparison to standard curve generating use known genome equivalent or nucleic acid harvested from the titered virus control. This is application we use it in molecular microbiology lab. In genomic profiling assay, the genome is analyzed for a duplication or deletion assay. Or deletion assay design focus on efficiency and the accuracy of discrimination of single copy deviations. And we can use it in allele discrimination assays. We can detect the variation down to the single nucleotide level, and point fluorescence is measured. Thank you for your listening. Thank you for your attention during my presentation. I know this is very long actually, but still we have the most important part to cover, inshallah, part 2, whenever you want. Thank you for your listening again, and I'm happy to have your questions to answer.
يعطيك العافية دكتورة سحر، مرة ما شاء الله كونتنت جميل، وأكيد إنه فاد ناس كثير، واللي اشتغل في اللاب مرت عليه أشياء كثيرة زي كذا، فشكرا لك على وقتك وشكرا على البرزنتيشن. إذا في أحد عنده أي سؤال either raise hand أو الشات.
I'm looking for a question actually. Inshallah, if anything it is not clear or you would like to understand it more, we can incorporate it in the second part, inshallah, next week. It will be so, I don't know if it is next week or not. Dr. Reem, she will let us when we can presented, but hopefully it will be also it will be beneficial part for molecular genetic diagnostic. Waiting for your question actually. Unfortunately, I can't see your comments. Thank you for you to actually.
شكله غالبا ما في أسئلة حاليا. طبعا أنا حطيت رابط للتقييم حق المحاضرة. إذا في أي فقرة أو بارت تبغون يتغطى بالبارت 2 ممكن بس تضيفونه لنا في الكومنت.
ممتاز.
ويعني راح نحاول الدكتورة سحر عشان نتأكد إنه موجود في البارت 2 إذا حابين. وشكرا لكم، شكرا لوقتكم، شكرا لك دكتورة سحر على وجودك.
معذرة الوقت متأخر بس إن شاء الله إنه استفدتوا يعني. And I think the lecture it's recorded already. You can يعني back to it at any time. And also I'm very welcome to share my slides after the part 2 inshallah. I will leave my email as well with Dr. Reem. If you need any help, you have any queries, we missed something or it's not clear, you are very welcome.
دكتورة سحر في one question in the chat.
سمي.
في question في الشات.
طيب فضلت محاضرة. طيب in case of Fragile X syndrome, what's the best technique you have? ألو؟
إيه موجودين معاكي.